Welcome friends, myself Shoptapurni from BioLive. So today we are going to discuss about a very important topic of recombinant DNA technology, blue-white screening. So first of all, what is the purpose of doing blue-white screening? See, in recombinant DNA technology, we try to understand if our host cell is getting transformed with the recombinant DNA containing cloning vector or not. Okay, so for that purpose we need to do the blue-white screening. So see, this is the basic of cloning method. So for cloning you need a cloning vector. This is a cloning vector and this is the restriction enzyme cutting site. Okay, this is nothing, this black is nothing. Okay, this is the restriction enzyme cutting site. And this is your target DNA or foreign DNA, and these are the restriction enzyme cutting site of your target DNA. We know that we cut both cloning vector and the target DNA with the same restriction enzyme at the same site or at the same palindromic sequence. Okay. Now, two things may happen. Either this cloning vector will incorporate this target DNA and form a recombinant DNA, or it may not incorporate the target DNA and its end will stick with each other and form a non-recombinant vector. Okay, so there will be two things, either recombinant vector or non-recombinant vector. Now you need to transfect your vectors, your cloning vectors into the host. So now introduce your vectors into the host. So here three things may happen. This is your host, your bacterial host. It is the bacterial DNA. Either your host will get transformed with the recombinant DNA, okay, with this one, or it may get transformed without the recombinant DNA, okay, that means with non-recombinant DNA, or may not incorporate the cloning vector at all. Okay, these are the three things that can happen after the transfection. So now you need to select which of your, in the colony, in the Petri plate, which colony contains the transformed cell or transformed host with recombinant cloning vector. Okay, so you need to find out this one, and for this we use some screening or screenable markers that produce visible color. Okay, and so that we can easily determine which host or which colony has got transformed with the recombinant DNA. So in blue-white screening method, we are using some screening markers or screenable markers which contains reporter genes. Okay, like here the LAGZ gene we will discuss briefly. Okay, here the LAGZ gene which will produce an enzyme like beta-galactosidase enzyme which generates color, the blue color, reacting with the substrate. The substrate is X-gal here. Okay. So see here, what is the principle behind this blue-white screening method? The principle is alpha complementation of mutant beta-galactosidase gene, that is LAGZ, of host bacteria E. coli. Okay, so what does this statement mean? See, in the wild type of host, that means the E. coli host, they contain wild type of LAGZ gene. Okay, we know LAGZ gene is present in lacoperon, and LAGZ gene produce or encodes beta-galactosidase enzyme, which is active when it is a tetramer. This beta-galactosidase enzyme breaks down the lactose into glucose galactose, or it may also break down an analogous substrate of lactose known as X-gal into galactose and 5-bromo-4-chloroindoxyl. Okay. Upon breakdown, these 5-bromo-4-chloroindoxyl gets dimerized and oxidized and form 5,5'-dibromo-4,4' which gives blue color. Okay, so if the host is wild type, it will form blue color. Okay, it will react with X-gal and will form blue color. Now, what happens if we introduce a plasmid vector into this host or into a mutant host? Let's see. So see, if we take a mutant strain of E. coli or our host, that is known as LAGZ delta M15 mutant, where LAGZ gene is mutant. Okay. So this mutant LAGZ gene will produce omega peptide, not your beta galactosidase enzyme. It will produce omega peptide which lacks 11 to 41 amino acid at its N-terminal end. Okay, so as a result, it is inactive and does not form the tetramer. So see here, it is a truncated kind of protein. Okay, so now if we introduce a plasmid carrying LAGZ alpha sequence, that is non-recombinant plasmid, what will happen? And we will see if we introduce the same plasmid with the target gene or the recombinant plasmid, then what will happen? See, this is our plasmid. It contains LAGZ alpha gene, okay, and it has a multiple cloning site, but no targeted gene or no foreign gene is introduced here. So this LAGZ alpha gene will encode a 59 amino acid, the first 59 amino acid of the N-termini of normal LAGZ gene. Okay, so here they are lacking 11 to 41 amino acid and they are producing 59 amino acid at the same N-terminal end. So obviously this peptide is known as alpha peptide, and in combination with omega peptide and alpha peptide will produce functional beta galactosidase. See here, this part is alpha peptide, this part is omega peptide, and they are now producing functional beta galactosidase which will break down the X-gal into a blue component. Okay, so what you will get from here, that if you introduce a non-recombinant plasmid containing intact LAGZ alpha gene which complements the omega peptide, then you will get the blue color, as because you are getting the functional beta galactosidase gene. Okay, but what will happen if the LAGZ alpha sequence of the plasmid is being cut at the multiple cloning site and is recombined with a targeted DNA? Okay, so see here, the same cloning vector containing LAGZ alpha gene have a multiple cloning site, and where there is a cut and target DNA has been introduced here. As a result, what happens? The LAGZ alpha gene, this gets disrupted. So no alpha peptide will be produced. So if no alpha peptide is produced, what will happen? The mutant host where omega peptide is produced, it will not get combined with the alpha peptide, and as a result no alpha complementation will be there. As a result no functional beta galactosidase will be there, and as there is no functional beta galactosidase, no one can break the X-gal and form the blue color. So as a result you will get white colony. So from here we can say that if you introduce a plasmid with a targeted gene, that will disrupt the LAGZ alpha gene of the plasmid and no beta galactosidase will be produced then, and as a result white colony will be formed. In a Petri plate, if you find two types of host cell colonies, that is, one is blue colony and another one is white colony, what you can interpret from that? See, blue colony means they are transformed host cell, but they contains non-recombinant vector. Okay, so they are transformed host cell but without recombinant DNA containing vector. That means they are transformed host cell, they have incorporated the plasmid, but plasmid have not incorporated the foreign DNA. And if you get white colony, you may interpret that they are transformed host colony with recombinant DNA containing vector. See here, they are the transformed host with a recombinant plasmid. Okay, that means here in this plasmid or in this cloning vector, the foreign DNA has been incorporated. As a result, the LAGZ alpha gene got disrupted and no functional beta galactosidase can be produced and X-gal cannot be broken down into blue component. Okay, So from these two colony you can easily say that which colony contains recombinant DNA and which colony does not contain the recombinant DNA. Now another question may arise in your mind that the non-transformed cell, what will happen to the non-transformed cell? That means those hosts which have not incorporated plasmid at all. Okay, they also have omega peptide, but as they have not incorporated the plasmid, they don't have the alpha peptide, so they can also form the white colony, right? But no, to omit this confusion, we have to select which cell got transformed and which cells did not got transformed before doing this blue-white screening. Okay, so at the first step, like that is at this step, when you are getting 3 types of cells, just after this step you need to do another method to divide these 2 cells from this one. Okay, so that the non-transformed cells will be at one side and this transformed cell will be at another side, and then between these 2 cells you will screen for the transformed cells with recombinant DNA. Okay, so that is all about today's video. I hope this video will be useful for you, and if you find this video useful, don't forget to like and share it with your friends, and don't forget to subscribe to my channel. Thanks for watching and happy learning.